All right, hey everyone. This is your set of notes for nucleic acids. So here we go. Nucleic acids are a polymer. Again, poly means many. So these are your biggest units. We'll talk about the monomers here in a second. But your two nucleic acid polymers are DNA and RNA. And we'll cover both of these in more detail later, but we're just kind of introducing them for now. The nucleotide is the monomer for nucleic acids. The nucleotide is the monomer. So let's look at the anatomy of a nucleotide. Nucleotides have three components. They have a phosphate group. Again, you should always know that phosphate's PO4. It has a negative charge. This is why DNA is negatively charged. There's your phosphorus. So there is a phosphorus there, obviously, PO4. You have a 5-carbon sugar, which is right down here in more detail. So this is your 5-carbon sugar. Again, each vertex here is a carbon. We're not going to show you every single atom, but each vertex here is a carbon. And then the 5th carbon is actually right up here. We'll look at that here in a minute. And then the third component is your N base, your nitrogenous base. Your nitrogenous base. And there are 5 of these things total. Uracil is unique to RNA. DNA will have C, T, A, and G. But these are your options. The smaller ones, the ones with single ring structures, are just called pyrimidines. The larger ones with double ring structures are called purines. The width of a DNA molecule is always going to be 3 rings, right? Because if you have C and G, that's 1, 2, 3 rings wide. If you have an A and a T, that is also 1, 2, 3 rings wide. So the width of DNA is constant. Mutations are going to mess that up too. It'll change the width of DNA, but just kind of know this for right now. This is pretty basic. This is going to be a quick set of notes. So I'm gonna keep going. If you look at the 5-carbon sugar options, you have 2 options. If you have DNA, that 5-carbon sugar is deoxyribose. That's where the D comes from. And if you have RNA, that sugar is ribose. That is where the R comes from in RNA. And the only difference, if you're looking at these, you can pause it and find it if you want, or you might have already seen it. The only difference is DNA has a hydrogen off this carbon number 2, whereas RNA has a hydroxyl group off this carbon number 2. I am going to draw out this really quick just to show you how the carbons are counted. That's what these numbers are here for. So again, you have a 5-carbon sugar, so I'm going to draw a pentagon. Now, not every point here is actually a carbon, because this top one is actually an oxygen, right? And you actually start your numbering convention at the oxygen, and then you're going to number them clockwise. And you might remember this from chemistry. So this first carbon here is carbon number 1. This carbon here is carbon number 2. This carbon here is carbon number 3. This is carbon number 4. And the 5th carbon is actually outside the ring structure. 5th carbon is actually out there, right, all the way out here. So that's how we get the numbers. We're going to pay special close attention to carbon number 5 and carbon number 3. And this is why. When we join these together, those are the 2 carbons where the reaction occurs. We're going to do this, or your enzymes will do this, just the same way with proteins and carbs. You're going to have a dehydration reaction that removes water. So on the left here, I'm going to split this. We're going to look at the left-hand side first. You've got 2 nucleotides already linked together, and we're down here, and we want to add this 3rd nucleotide. So what happens is the OH group on the 3' carbon and a hydrogen from the adjacent nucleotide's phosphate group is where the water comes from. You form a covalent bond between the oxygen in the phosphate group and the 3' carbon, right? So you're forming this bond right here. And now you have the nucleotides linked together, right? That's dehydration synthesis. Let's see, honestly, I think that's it. So here we go. Details will come when we talk about DNA and RNA in a future unit, but that is the basics for nucleic acids. Do pay attention to the 3' side. It's the end of the molecule that has this 3' carbon kind of hanging off by itself. And the 5' side. It's the side of the DNA or RNA that has the 5' carbon hanging off by itself. Again, we'll pay close attention to those later. But there you have it. I hope that helps, and I'll see you next video.